In this video, we will discuss about some of the combination CAR T and CAR NK immunotherapies that can currently be used to diagnose patients who are affected with cancers. So, some of the drugs are being used to prevent the suppression of activated immune cells, or we can say that they they belongs to this immune checkpoint blockade therapy. So they will blockade the receptors on the immune cells that can interact with the tumors and leads to the suppression of the immune response. So some of these drugs are this tocilizumab that can interact with this IL-6 receptors, novolum, novolumab can interact with this alpha PD-1, and this azitulizumab can interact with this PD-1 ligand that are expressed on the tumors. They were preventing the interactions of PDL1 and PD-1 receptors, and the suppressions of the immune response will be reduced. So these are some of the blockade monoclonal or nano antibodies, and some of the activated antibodies are also being used, and they can be used to activate the immune cell response. For example, the beads that has anti-CD3 and anti-CD8 uh, anti-CD3 and anti-CD8 and antigens, so it can interact with the receptors, anti-CD3 receptors or anti-CD8 anti-CD28 receptors, thereby helping the activations by triggering the intercellular signalings within the immune cells. The other activating monoclonal and nano antibodies are this alpha 4 1BB. It can also lead to the co-stimulatory molecules that can target the tumor cells. And right hand side shown is about the CAR NK cells, and left hand side is about the CAR T cells. So the CAR NK cells also this nivolumab can also be used, and azitulizumab can also be used to prevent the ICB. And this ANKR can be overexpressed. ANKR can be overexpressed, or this KIR can be blocked, thereby this lenalidomide, it will lead to the overexpression of the ANKR on the cell surface, and this will lead to the enhanced anti-tumor response against the tumors. At the same time, uh, the suppressions of the immune cells can be reduced through this KIR blocking monoclonal antibodies, as can interact with the KIR receptors. And the oncolytic virus related CAR NK cells that can be used through this uh, by expressing this CAR construct on the CAR NK cells, and that can, once it is activated, it can introduce the co-stimulatory response such as alpha EGFR and IL-15. And the other things are this immune engager. This immune engager, what it will does is that it can interact with solid tumors. What it will immune engager will does is it will lead to the release of this antibody dependent cell cytotoxicity. So it can gather the cell tumor specific antigen informations, and thereby helps in generating the tumor response specific to those solid tumors. And these are on the left hand side the immune engager in the CAR T cells, where it does the same thing, that is, it takes the antigen informations from the solid tumors and develop antibodies against those solid tumors. This OV19 is about the oncolytic virus in the CAR T cells, where this OV19 will lead to the truncation of CD19, thereby exposing them to the CAR T cells. And the lenalidomide is can also act as an immune modulator by increasing the proliferation cytotoxicity and cytotoxin release in the CAR T cells, that can increase the immune efficiencies against the solid tumors. The other kind of CAR constructs that are used is viral CAR transductions. So in this case, the CAR has been transduced using adeno-associated virus thereby leads to the expression of thereby leads to the expression of uh, thereby leads to the expression of the gene of interest inside the virus. So it can be done either by adeno-associated virus, lentivirus, or retrovirus. And once the gene of interest is within the virus, it can be fused inside the cells and integrate with the uh, cells machinery. That will lead to the transcription and translations, and also on the expansions of this virus to the other neighboring tumors. And this LV serotypes transduction efficiencies are increases from SVG to BAEE EV. The other car engineerings that people are used right now is about the electroporation, where mRNA are being electroporated. Car related mRNA are being electroporated, and thereby that it can use the cells machinery for translation. And this can be used in the form of this nanovector DNA, where car construct are being electroporated inside the nucleus, where it will be transcribed and translated, and it will lead to the base editing, and it can also be used for this CRISPR-Cas9 system. So, and there's the one things that is currently being used is about in situ alpha CD3 and alpha CD28 expansions, and that are done or achieved using mCAR-T injections, and this seems to be uh, and the results of this approach is prominent at this stage. The biomaterial can also be used, and this is just a idea right now. It can also be used for in vivo targeted payload delivery or CAR delivery to the unedited lymphocytes. CAR delivery to the tumor sites. CAR delivery to the tumor sites, not unedited lymphocytes. These are some of the approach where CAR T cells and CAR NK cells therapies can be combined to increase the efficiencies of immune response against the tumors. That's all. So that's all for this video. If you need to understand more about these experiments and not experiments, these ideas in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.